Hello students, welcome back to our YouTube channel Food Tech Online. So today we'll be covering MCQs set number 7 for ICAR-PG Food Science and Technology. So let's begin. Before moving to the questions, let me quickly remind you about our ebook which is related to Food Science and Technology ICAR-PG, and it contains the courses such as food chemistry and nutrition, food microbiology, food products technology, food engineering and more. The link of this book is given in the description box. Question number 1. Which chemical is released in response to allergen in the body? Options are option A histamine, option B epinephrine, option C thyroxine, option D serotonin. And the correct answer is option A histamine. It is a nitrogenous compound and it is a chemical messenger that normally acts in the body with various functions such as inflammatory reaction, allergic reaction, uh, neurotransmitting action, etc. It is released whenever an allergen is detected by a body as an immune response. The others are also some of the hormones as well as neurotransmitter. For example, serotonin is an also called as happy hormone, and it mediates the sense of satisfaction, happiness and optimism. It also involves the other functions such as mood, in cognition and the memory, learning, vomiting, etc. Epinephrine is also commonly called as adrenaline, and it is secreted by the adrenaline gland. It is also a neurotransmitter as well as a hormone and plays important role in life-threatening conditions by pumping more blood to the heart so that uh, there is no loss of blood, and it also decreases the sensitivity to pain. Thyroxine is also a thyroid hormone which is secreted into the bloodstream via thyroid gland and plays important role in the metabolism, heart and muscle function, brain development and bone maintenance. It is used in the treatment of hypothyroidism where the symptoms such as weight gain, tiredness and depression can occur. Question number 2. What is the use of sodium bicarbonate as a humectant, preservative, leavening agent, or stabilizing or emulsifying agent? So the correct answer is option number C as a leavening agent. So sodium bicarbonate, formula of which is NaHCO3. This, when it comes in the contact with the acidic environment, it will break down into components like the carbon dioxide and water and other components. So water will further convert into steam, and both will give the rising effect. So this is specially used in the bakery industry to rise the dough, and it is also used in the beverage industry to aerate the or to carbonate the drinks. So it is also used as a pH regulator and flavor and for flavor enhancement. The other agents like the other additives like the humectant, it is used especially in the powdery material or granular materials to keep them flowy, uh, or to keep them free flowing. Preservatives are used to extend the shelf life of the food. It can be chemical preservatives, for example, the salt, sugar, and oil, or it can be other chemical preservatives like the sodium benzoate or sorbic acid, etc. The stabilizing agents are those which stabilize any type of mixtures, and it will retain their physical properties. Emulsifying agents are used to mix two type of immiscible liquids, for example oil and water. The classic example of emulsifying agent is the lecithin. Question number 3. Which method is used for alternating environmental conditions in which spoilage microorganisms are unable to grow in the food? Options are preservation, fortification, sterilization, and sanitation. Correct answer is option A, preservation. So, in this preservation technique, we have different methods. Like we can use heat, we can use freezing technique, or we can add some chemicals or the food additives to alter the environmental conditions so that microorganisms, any kind of microorganisms like yeast, mold and bacteria, it won't grow back, and this will extend the shelf life of the food. Then we have the fortification. So fortification is adding the basic vitamins and minerals which are lost during processing, and these are added back to the most commonly consumed food items such as the wheat, rice or the other cereals. Then we have milk, Then most of the vitamins and minerals which are added are the iron, and we have vitamins like vitamin A, etc. So this fortification is needed so that there is prevention as well as the control of the micronutrient deficiencies. Then we have the sterilization. So it is a process where most of the microorganisms are either inactivated or killed using the temperatures, or it can be the heat and steam and chemicals to destroy, as well as their spores are also destroyed. So that is sterilization. Then we have sanitation. It is a process where pathogenic microorganisms, so there is a difference between the sterilization and sanitation. Here, most of the microorganisms plus their spores are removed, and whereas in sanitation, we remove the only pathogenic microorganisms. And sanitation is mostly done of the equipment or the processing environment, and it removes 99.99% of the microorganisms. So here most of the viruses and the spores are not removed, but this process reduces the chance of foodborne illnesses. Question number 4. The transfer of microbes from one food to another, from equipment to food, or from people to food is known as. Options are contamination, spoilage, cross-contamination, and poisoning. Correct answer is option number C, cross-contamination. So cross-contamination is an unintentional transfer of chemical, microbiological, foreign particles or allergens from people to food, or from dirty surfaces or equipments which are used in the food preparation, or from one type of food to another. So this can be avoided by taking proper measures and by training the food handlers to identify and prevent the potential hazards. Then contamination, it refers to improper handling of the raw and the cooked food, uh, which can contaminate the products with diseases such as bacteria, with disease causing organisms like the bacteria, parasite, and viruses. So the causes of this is improper handling, storage, and food preparations, and prevention can be using proper equipments and sanitizing the surfaces. Food spoilage, it is a process of change in the physical, chemical, and microbiological properties of the food, and when these properties are altered, it makes the food unfit for the consumption. And this is caused by physical changes, like if the food is being damaged, then chemical changes, like there is increase in rancidity or increase in the pH. Then enzymatic spoilage, that is the browning reactions, and the microbial factors also destroy the food, or it gets spoiled. Then food poisoning is caused by food which is contaminated by the microorganisms, and some of the microorganisms deposit the toxins, and which may cause the symptoms like the nausea, vomiting, and diarrhea. Question number 5. What happens to the can of canned foods at high altitudes? Options are: it gets contracted, it gets expanded, there is no change, or microbes are destroyed. So the correct answer is option number B, it gets expanded. At high altitudes, there is a atmospheric pressure difference which is created. The atmospheric pressure drops with the height. As the height increases, or as there is elevation, the atmospheric pressure drops, and hence the canned products can expand at high altitudes. Question number 6. The term applied to stomach and intestinal disturbances caused due to ingestion of food contaminated with microbes or their toxins is. Options are: food allergy, food poisoning, food infection, and food spoilage. Correct option is food poisoning. So food poisoning is also called as food intoxication. Food intoxication. So this food or intoxication happens when a person consumes food or beverages that contain these type of toxins which are already produced by the microbes, especially the bacteria. The bacteria which produces the toxins are, for example, Staphylococcus aureus, Salmonella, Clostridium perfringens, Campylobacter, etc. The Salmonella is having a very common, I'll write it here, Salmonella is most common which causes the food poisoning, and the toxins which are produced are endotoxin and exotoxin. Endo and exotoxin. Then the food infection is also called as foodborne illnesses. This is also called as foodborne illnesses. And these foodborne illnesses are caused by the bacteria or any type of microbes, but these are directly consumed through the food and beverages. That means the bacteria itself, the microbes itself are directly consumed through the food and beverages. So these both of these, the poisoning as well as the infection, both of these cause the symptoms such as nausea, vomiting, and these are very fatal. So these can even cause death, and these can also be caused by viruses, etc. In the food infection can also be caused by viruses, especially the rotavirus, rotavirus and hepatitis A, rotavirus and hepatitis, hepatitis A. Then we have the food spoilage. Now, food spoilage can be it is a process of change in the physical chemical properties of the food, and it makes it unfit for the consumption. So these can be caused by the physical factors, chemical, enzymatic or microbial factors. For example, if it is a physical factor, then it can be the damages of the surface. Then if it is a chemical factor, the food can be spoiled, and after spoiling the food, it will increase in the acidity, or it will give off flavors, etc. Then food allergies. Food allergies are caused, it is a autoimmune or it is an immune response to some kind of food. The cause is unknown, but still uh, our body identifies it any particular food as a threat, and in its response a histamine is released, and the most commonly found allergies include the foods such as the milk, fish, egg, peanut or any type of nut, etc. Question number 7. What is the importance of freezing of food? Options are to kill bacteria, to seize multiplying bacteria, to preserve vitamins, and to kill virus. Correct option is option number B, to seize multiplying bacteria. So during freezing, the physical state of the matter is changed, where the energy is removed during the cooling below the freezing temperature. So at extreme temperature, it retards the microbial growth and slows down the chemical change, which might affect the quality and cause the food to spoil. So the freezing of during the freezing of food, most of the bacteria will retard their growth. That is, to they cease the multiplication. But as soon as they get the proper temperature, they can still survive. Question number 8. The danger zone in the food preparation refers to the temperature range from, options are 0 to 40 degree Fahrenheit, 40 to 140 degree Fahrenheit, 140 to 180 degree Fahrenheit, and above 180 degree Fahrenheit. Correct option is option number B, 40 to 140 degree Fahrenheit. So the bacteria mostly grow rapidly in the temperature range of 40 to 140 degree Fahrenheit, and they double in no time. It can take only 20 minutes to double this type of bacteria in this type of temperature range. So the bacteria like the Staphylococcus aureus, Salmonella, E. coli, Campylobacter, these are very harmful bacteria, and these might cause severe kind of diseases. So the harmful bacteria can be the E. coli, Salmonella. Okay, so to avoid the growth of these type of bacteria, the food is labeled as in the danger zone. that is this for 140 to 140 degree Fahrenheit temperature range. So if you keep the food which is not properly cooked or undercooked at a room temperature for too long, that is more than 2 hours, then the food can have the growth of these type of bacteria. So the hot food should be kept hot at a temperature range of 140 degree Fahrenheit and above. And if you're freezing the food or cooling down, then you should maintain the freezing temperature below 40 degree Fahrenheit. Question number 9. Technology which is involved in the manufacture of frozen pizzas. Options are freeze drying, IQF, which is the individual quick freezing. Then third option is freezing, and last one is the coating. So here the correct option is option number B, that is IQF, individual quick freezing. So it is a process in which the uh, it preserves the individual pieces of food instead of clumping them together in a one large block as the traditional method does. So this method keeps the food fresher for a longer period of time because it prevents the ice crystals for forming on the surface. For example, um, fluidized bed dryer is used to dry the smaller pieces of the fruits and vegetables. I'll write it here. The peas, then corn, these type of vegetables are dried using this IQF technique. Then we have got freeze drying. This is a method wherein the water is in the form of ice is removed under low pressure from a material by sublimation. So initially the food product is freezed completely and then dried under low pressure. Sometimes vacuum is also applied. This method is also called as, this freeze drying technique is also called as lyophilization. Lyophilization, or this is also called as cryodesiccation. Cryo, cryo means freezing thing, and desiccation means drying. So cryodesiccation. And this technique is especially applied to fruits and vegetable as well, and it enables the growth of pathogenic microorganism. It keeps the food fresh without cold chain and maintains the shape and size of the food, because in traditional dehydration, uh, mostly the fruits and vegetable, they shrink in size. So here that is the advantage. And this method also retains the nutritional as well as sensory properties. Then we have got freezing. It is a technique in which uh, physical state of the matter is changed as the energy is removed during the cooling below the freezing temperature. So extreme cold temperature retards the microbial growth and it slows down the chemical change. And the storage temperature for frozen food is minus 18 degrees Celsius. Question number 10. Alternate term for cold sterilization is, options are pasteurization, irradiation, freezing, and blanching. Correct answer is option number B, irradiation. Uh, Irradiation uses ionizing radiation such as the gamma radiation, which inactivates foodborne illnesses or foodborne pathogens without the heat. That's why it is called as cold pasteurization or sterilization, depending upon the dosage of the radiation used. So this is specially suitable for the heat sensitive items, and it has various benefits, or it has got various benefits, especially in terms of it can reduce the sprouting in case of onions and potatoes or garlic. And this uses gamma radiation, and it is especially used to stop sprouting. Then it is used to decontaminate the spices and herbs. Decontaminate decontaminate spices and herbs. Then it reduces the microbial load. Then it delays ripening in case of mangoes, bananas, and papaya. Then we have got the blanching technique. Now blanching stops the enzyme action which otherwise cause the loss of flavor, color and texture. It also removes the surface dirt, microorganisms and brightens the color. It slows down the vitamin loss. It also removes the air and it softens the product. That's why it is a prior step before any type of food processing like freezing, dehydration or canning. And it is specially used in fruit. It is specially used for vegetables, and the temperature used for blanching is it is less than 100 degrees Celsius. Then we have got pasteurization. Now pasteurization also is a technique to reduce the pathogenic microorganisms pathogenic microorganisms and this also uses less than 100 degrees Celsius temperature to control the pathogenic microorganisms. Then freezing is a technique wherein we freeze any material uh, because it reduces or it retards microbial growth. It retards microbial growth, and the temperature used is less than minus 18 degrees Celsius. That is the freezing temperature. Question number 11. What is the time temperature combination to treat milk at high temperature short time? Options are 63 degrees Celsius for 30 minutes. Option B is 72 degrees Celsius for 50 seconds, 89 degrees Celsius for 1 second, 90 degrees Celsius for 0.5 seconds. And the correct option is option number B, 72 degrees Celsius for 15 seconds. So at this temperature, the sensory properties like the flavors and the nutritional quality like some of the vitamins, these are still retained, and this method is also called as flash pasteurization. This is also called as flash pasteurization. So and the short form of this is HTST. And this another method of pasteurization where it is also called as low temperature long time method. This uses 63 degrees Celsius for 30 minutes. Since the time is very long, which is 30 minutes, the flavors and some of the vitamins are lost. So this is a very preferred method, HTST. Question number 12. SOP stands for options are standard operating procedure, small outline package, standard of practice, none of the above. And the correct answer is option number A, standard operating procedure. So standard operating procedure is the set of step-by-step instructions which are compiled together by an organization to help the workers to carry out their routine operations. It may include the hazard identification or how to go about the working environment. Then SOPs are aimed to achieve efficiency, quality output, and uniformity of the performance while reducing the miscommunication and failure to comply with the industry regulations. So, especially where the safety analysis is involved, the hazards are to be identified and control methods are to be described. So, these are specially included in the SOPs of the food industry. Question number 13. Cutting boards must be sanitized with options are soap, chlorine, bleach, detergent, and none of the above. Correct answer is option B, chlorine bleach. So it is a strong and effective against most of the wide range of microorganism as it contains active ingredient called as sodium hypochlorite. Sodium hypochlorite. So it effectively removes and it is a very strong disinfectant, so it kills most of the microorganisms. Thus it is used to clean the cutting boards or the chopping boards. Question number 14. The receiving temperature of chilled food should be at or below the temperatures. Options are 7 degrees Celsius, 5 degrees Celsius, 3 degrees Celsius and 6 degrees Celsius. And the correct option is option number B, 5 degrees Celsius. The temperature which is below 8 degrees Celsius significantly slows down the bacterial growth, and the colder the environment, it is very harder for the bacteria to grow or multiply. So to practice the best temperatures, the chilled food item should be kept at kept at or they are received at a temperature which is 5 degrees Celsius. This is also 41 degree Fahrenheit. So this is the answer. Question number 15. HDHD with respect to pasteurization is, options are option A high temperature short time treatment, option B high temperature short time, option C high temperature storage test, option D none of the above. Correct answer is option B high temperature short time. Question number 16. Pasteurization is done to kill, options are pathogenic microorganism, all microorganism, viruses, yeast and spores. And the correct answer is option number A, pathogenic microorganisms. So pasteurization is a process where the food material is treated at a temperature less than 100 degrees Celsius, and the pathogenic microorganisms such as the E. coli, then Salmonella, then Yersinia, Listeria, these type of bacteria which are harmful and which cause diseases like the tuberculosis, Q fever, diphtheria, etc. These are killed. And the pasteurization is most commonly used in case of the milk, as we all know, and the fruit juices. Fruit juices. So some of the bacteria some of the yeast and viruses or its spores are not killed. Hence, pasteurization is only to kill the pathogenic microorganisms. Question number 17. What is the storage temperature of frozen food? And options are 4 degrees Celsius, 0 degrees Celsius, minus 18 degrees Celsius, and 10 degrees Celsius. Correct answer is option number C, minus 18 degrees Celsius. This is also in 0 degree Fahrenheit in Fahrenheit scale, and most of the bacteria sees their growth or their growth is limited at minus 18 degrees Celsius. Thus, this temperature has to be maintained. And this 4 degrees Celsius is the temperature for refrigerator or refrigeration temperature. And there is slight difference between refrigeration and frozen food, because refrigeration is it doesn't retard the growth of most of the microbes. It only limits the pathogenic microbes. At this temperature, even psychrophills can grow. Thus, 4 degrees Celsius is only the chilling temperature which is maintained to cool down the product. But at minus 18 degrees Celsius, the quality of the product is also retained, and there is retardation of the most of the bacterial growth. Question number 18. Elimination of all forms of microorganism is called as, options are pasteurization, sterilization, blanching and epitization, and the correct option is option B, sterilization. So sterilization is a process where we remove all forms of microorganism that is viable as well as their spores. Both are removed, and sterilization can be achieved using heat or steam or using chemicals in the form of gas. So that is sterilization. And pasteurization is elimination of only pathogenic microorganisms, and that is applied using the heat which is less than 100 degrees Celsius. Blanching is a process of it is not exactly aimed to remove the microorganism, but it has other advantages like it retains the color, it removes or it inactivates the enzymes, and that is why the blanching is done. So it is also done at a temperature less than 100 degrees Celsius. And epitization is also called as canning, and it is a process where we remove pathogenic microorganisms and their spores using heat as well as steam. And the cans are the processing of can is done using a sterile environment, and the cans are also sterile, and then the process after the process it is sealed also in a proper environment. So that is epitization or canning. Question number 19. What help in maintaining the pH? Options are option A, buffering agents, option B, sequestrants, Option C, anti-caking agents. Option D, foaming agents. And the correct answer is option A, buffering agents. These are the agents which are used to stabilize the pH, that is the hydrogen ion concentration of a solution, and these improve the flavor, increase the stability of the foods. These are the salts of weak acid and weak bases, and some of the examples of the buffering agents are bicarbonates, hydrogen phosphates, carbonates, etc. The sequestrants are the agents which prevent the adverse effect of metals, especially the iron, copper and nickel ions, which catalyze an oxidative breakdown of the food forming chelates, and these sequestering agents inhibit the decolorization and of taste and rancidity. Examples are the calcium chloride, sodium or potassium gluconate, etc. These are specially used in canned fruits and vegetables and as a salad dressing. The third is the anti-caking agents. These are added to powdered or granulated materials such as the table salt or the confectionaries to prevent the lump formation and for the ease of the packaging and the flowability. These function by either absorbing the excess moisture or by coating the particles, making it water repellent. Examples are the calcium silicate and sodium ferrocyanide. Antifoaming agents are also called as defoaming agents. These reduce the foaming caused by the protein or the gases that interfere with the processing, and these are especially used in confectionery, bakery products, dairy and alcoholic beverages, or the beverages such as syrups and molasses. And the antifoaming agents examples are the polyoxyethylene stearate and polysorbate propylene glycol, etc. Question number 20. What is the primary purpose of blanching of food? Options are to clean the food, then we have to prevent pest infestation, then inactivate the enzymes in the food, and to prevent food from drying. So the correct option is option C, to inactivate the enzymes in the food. So blanching is a process where mostly the vegetables or some of the fruits are blanched at a temperature less than 100 degrees Celsius, mostly in the range of 75 to 85 degrees Celsius, and this is aimed to inactivate the enzymes, because if the enzymes are still present, then they can cause discoloration or they can degrade the food. So to inactivate the enzymes, we do the blanching. So it is a pre-treatment or a preparatory method which is done before the major processing, and it also removes the occluded air or the oxygen which is present. So blanching can also be done prior to a canning method. Question number 21. What is added to fruits before freezing commercially to protect quality? Options are vitamin E, ascorbic acid, water and sugar, and the correct answer is option number B, ascorbic acid. During freezing, uh, the enzymes are only slowed down, their activity is only slowed down, and to maintain the quality, we have to retard their activity or completely cease their activity. And since most of the fruits and not fruits but vegetables are blanched beforehand before freezing, so that to remove the enzyme, fruits are very delicate in flavors and high in sugar and salt, thus they are not blanched, because after blanching there is an osmosis reaction and thus the salt and sugar is removed. So what else we can add to fruit is we can add vitamin C or ascorbic acid. Ascorbic acid is also called as vitamin C, and this retards or slows down the activity of the enzymes. The enzymes in general causes oxidation reaction, and thus they can also enzymes can cause browning reactions. So to avoid this, we can add vitamin C or ascorbic acid. Water, sugar and vitamin E are not added before freezing. Question number 22. What is the ability to trace the history, use of location of an identity by means of recorded information? And in terms of the product, it refers to the origin of the ingredients, history of process applied, distribution and location of the product after delivery. And the correct answer is option number C, that is traceability. So Traceability, it is the process for it is an ability to trace all the processes right from the procurement of the raw material to the production, consumption, disposal, to clarify when and where the product was produced and by whom. So this is important to improve the quality. So traceability is important to improve the quality, and to safeguard the product quality and safety. And then we have the nutritional labeling. So this is important because nutritional labeling should include what is the nutrient content of the food, and nutrient content means it should include the energy, which is given mostly in the calorie or kilojoules, and then we have got fats, then proteins, etc. So all the contents, all the nutritional contents has to be maintained on the nutritional label, and these are intended to use to guide the consumer in food selection. Then we have got the best before date. It gives the idea of how long the food will last before they change the quality. And the use by date indicates the food must be consumed before this date. So this is food must be consumed before this date. Before this date. And the best before is how long product, how long the product will be fit to consume. Fit to consume. Question number 23. Which of the following is an essential amino acid? Options are lysine, alanine, tyrosine, and serine. And the correct option is option number A, lysine. So the essential amino acids are these are the acids which are not synthesized by our body and thus should be incorporated through our diet because they are indispensable because they are indispensable nutrients. So there are 9 or more than that, that is 11 amino acids, but the common ones are the 9 essential amino acids. uh, those are the histidine, isoleucine, leucine, lysine, methionine, tryptophan, phenylalanine, threonine, and valine. So out of this, the lysine, which is given here, is an essential amino acid, and this lysine is an amino acid which is a precursor to many proteins, and its function includes treating or preventing the cold sores, reducing anxiety, and preventing and it prevents wound healing. So lysine is important, and the other which are mentioned here like the alanine, tyrosine, and serine, these are non-essential amino acids which are synthesized by our body. Question number 24. Canning is also referred as options are sterilization, pasteurization, apodization, and none of the above. Correct answer is option C, apodization. This is known as apodization because it was developed by a scientist called as Nicholas Appert, and it was named after him. So canning is a process where we remove most of the microorganisms as well as their spores, and it is done to increase the storage of the food. It involves the sterilization of food in an airtight container followed by hermetic sealing of the cans. Then we have sterilization. It is a process where there is complete destruction of the microorganism, and the process involves heating, that is heating initially, then we hold it and for a certain period of time, and then we cool it. Okay, so this can be done at a temperature at least 121 degrees Celsius for 15 minutes. And then we have pasteurization. It is done at a temperature less than 100 degrees Celsius, and it ensures the elimination of the pathogenic microorganism, mostly done in case of milk and fruit juices. Question number 25. Enzyme that converts starch to maltoses
Options are option A maltase, option B diastase, option C invertase, option D hydrogenase. And the correct answer is option B diastase. So diastase is also called as alpha amylase, and it converts starch into maltose, whereas maltase is an enzyme which converts maltose to glucose. And the invertase enzyme, it converts sucrose, which is also a disaccharide, into its two units, which is the glucose plus fructose. And the hydrogenase is an enzyme that catalyzes the conversion of molecules, that is H plus ion, to protons and electrons by heterolytic splitting mechanism.
So we have come to an end with the all 25 questions for the MCQ series, and this is our application Foodtech Online. It covers all the major examination, that is the FSSI, the GATE exam, the state FAOs, with the subjects like the food processing, food chemistry, food engineering, food microbiology, etc. Join our application, link of which is given in the description box. Thank you students for joining, and please subscribe to my YouTube channel and also join us on the social media, link of which is given in the description box.